In this video, we're going to look at the vertebrate neuromuscular junction. As the name suggests, the neuromuscular junction is the synapse that occurs between the motor neuron and the skeletal muscle fibers. In vertebrates, the cell body of the motor neuron lies in the central nervous system. It can lie either in the spinal cord or in the brainstem. Either way, the axon of that nerve cell will then exit through the spinal nerve or a cranial nerve and travel out to the target cell, the muscle fiber. We know more about the neuromuscular junction than about any other synapse in the body, in part because it's easily accessible, but also because it's implicated in a number of diseases. It is important to remember, however, that the neuromuscular junction is not a typical synapse, and it has a number of unusual features. One unusual feature is that the target cell, the muscle cell, is innervated by only one presynaptic cell. So the cartoon that I'm showing you shows that a single nerve cell can branch and terminate on several different muscle fibers, but any one of those muscle fibers gets input from only one presynaptic cell. There's only one neurotransmitter at the synapse, it's acetylcholine. There's only one neurotransmitter receptor expressed on the target cell, it's the nicotinic acetylcholine receptor. And there's only excitatory input, and what I mean by that is that the target cell is depolarized when the synapse is active. In addition to that, this is a very strong synapse, and what I mean by that is that a single action potential in the presynaptic cell is sufficient to bring the target cell to threshold, and we'll see that this is very different in central synapses. And finally, there's an unusual morphology. Let's begin by looking at that unusual morphology. So here's a little bit of a muscle fiber. Each one of those long pink cylinders represents a different muscle cell. And we can see axons coming up then and terminating onto the muscle fibers. And you can see that each one of the pink cells is getting only one input here. It's this end plate. Now, if you look closely at the end plate, you'll see that there are a lot of terminal boutons. But what you should know is that each one of those boutons comes from a single axon. So the axon might branch and have multiple points where it talks to the muscle cell, but there's only one neuron talking to a muscle cell. I want to talk about the different features that are present at this junction, and to do that we'll look at a cartoon. So here's a cartoon with the axon terminal of the motor neuron, and down here's the muscle cell. I'm going to blow this up even further so we can look at some of the players. Well, as expected, the terminal is packed with synaptic vesicles, and because at the vertebrate neuromuscular junction the neurotransmitter is acetylcholine, I've labeled this packed with acetylcholine. In the membrane of the presynaptic cell, we see active zones, and this will contain T-snares and the voltage-gated calcium channels that will be important for transmitter fusion and release. The postsynaptic cell has postsynaptic densities, where among other proteins we find the nicotinic acetylcholine receptors. And then if you look at the surface of the muscle cell, most of the muscle cell plasma membrane's smooth, but opposite the motor neuron, there are these deep folds in the membrane called junctional folds, and they align right at the spot where the neuron makes a contact onto it. These junctional folds, as well as the entire surface of the muscle cell, are lined with a special kind of extracellular matrix called the basal lamina, and this is going to provide structural support, for example, allowing it to maintain this unusual folded structure, and it will also anchor some proteins so that they're held right at the spot where they're needed. This is an electron micrograph of a neuromuscular junction, and at the top we have the cell, the nerve terminal. This nerve terminal contains many mitochondria. Remember, it's expensive to maintain the synapse, and in this region here there are numerous synaptic vesicles. The lower half of the figure shows us the muscle cell, and all of this structure down here you might recognize as the contractile apparatus. These are sarcomeres with actin and myosin. Here's a little bit of the membrane of the muscle cell, and I've highlighted in purple so that you can see the junctional folds and the fact that the membrane really is folded opposite the synapse. This is an image from a different neuromuscular junction, but once again we can see the axon terminal at the top and the muscle cell beneath it. I've highlighted the membranes. Here, because it's a higher magnification, we can see the numerous synaptic vesicles that are present in the terminal. In the muscle cell, we can see mitochondria here. And I think you can also see that there are these junctional folds. Now, this image shows us something else that wasn't maybe apparent in the previous image that I showed you, and that's what I've highlighted with this turquoise color. Notice that the space between the muscle cell membrane and the terminal membrane is not empty. There's a fibrous network in here. This is the basal lamina, that extracellular matrix that's lining the surface. If I take away the highlights that I've added so that you can see the actual micrograph, I hope you'll notice that the density of the muscle cell membrane isn't uniform everywhere. At the top of the junctional folds, we see quite a lot of black material, dense material. This is the postsynaptic density. And if you compare that to the base of a junctional fold, you'll see that the membrane there is less darkly stained, showing that there are fewer proteins there. There is also a concentration of proteins in the opposite cell, in the active zone, and we can see that here where the vesicles would be fusing. I want to walk you through the events that are involved in packaging and the release of acetylcholine at the neuromuscular junction. The transmitter at the neuromuscular junction is acetylcholine, and this is a small molecule. I've shown you its structure here. And this compound is synthesized in the terminal itself. If you think about it, this makes sense. The cell body for the neuron lies in the spinal cord. It might be, half a meter away from the site of release, and it would be inefficient to have to synthesize the compound in the cell body and then transport it all the way down to the axon terminal. So instead, the enzymes that are necessary to synthesize the compound are present in the terminal. Acetylcholine is made up of two building blocks, acetyl coenzyme A and choline. Acetyl coenzyme A may be familiar to you. It's an important intermediate in cellular metabolism. The cell is constantly making this compound in mitochondria. In contrast, choline is a more limited compound. We get it from our diet, although there are ways to synthesize it in the liver. But because it's not as commonly used in the cell will recycle this and reuse it at the site of the synapse. Synthesizing acetylcholine requires an enzyme. It's called choline acetyltransferase, and it runs to create acetylcholine from these building blocks. But once we've synthesized the acetylcholine, we need to concentrate it in the vesicles. People estimate that there are about 10,000 acetylcholine molecules in each vesicle, and they're at a much higher concentration inside the vesicle than they are in the cytoplasmic space. So that should make you think that the loading process is going to involve an active transporter. To load the vesicles with transporters, cells use a vesicular acetylcholine transporter called the VACHT. This is an antiport. It moves acetylcholine from the cytoplasm into the vesicle, and it pays for that movement by moving protons from the vesicle interior out into the cytoplasm. To establish the proton gradient, the vesicle membranes also contain a vacuolar ATPase pump, and this pump is running constantly, breaking down ATP and then moving protons into the vesicles so that there's a high concentration of protons. This will allow the antiport then to run and move the acetylcholine against its concentration gradient. Once the vesicles are loaded, then they need to be released. Remember that transmitter release is going to involve the arrival of an action potential at the terminal, influx of calcium, and then interactions between V and T snares. The transmitter can be released into the cleft, where it can diffuse across and interact with the postsynaptic cell, and the membrane itself will be endocytosed, and then it can be reloaded with neurotransmitter using the machinery I talked about earlier. Let's turn to look at the events in the muscle cell. Here's a blow-up of the muscle side of the synapse. At the top of the junctional folds, we'll have the receptors for acetylcholine. Again, these are nicotinic acetylcholine receptors. Nicotinic acetylcholine receptors are a type of ion channel. They're ligand-gated ion channels, or they're also called ionotropic receptors. These are membrane proteins that form pores. They're selected for a particular type or type of ions. The nicotinic acetylcholine lets through mostly sodium, but it will also allow through potassium and calcium. And these channels are going to open or close when a ligand, in this case the transmitter, binds to the channel. So here's our ligand gate. It's blocking the flow of ions through. Here's a binding site for the neurotransmitter. And in the absence of any neurotransmitter, the channel's closed. If we add acetylcholine, well, then the gate can flip into the open configuration, and sodium can move into the cell, depolarizing it. Thus, what's going to happen at the synapse will be that when the neurotransmitter is released, it's going to bind to these acetylcholine receptors, sodium will flow into the cell, and that can then depolarize the muscle cell. The muscle cell then, in response to the action potential in the motor neuron, can initiate its own action potential. And we would see that if we did an experiment where we stimulated the motor neuron to release transmitter and then recorded the voltage. What would happen is that after stimulating the nerve cell, the muscle cell itself would initiate an action potential. And ultimately, this is going to lead to contraction in the muscle cell. There's one more element present at the cleft that I want to talk about, and that's an enzyme called acetylcholinesterase. So this is an enzyme that can deactivate acetylcholine. Muscle cells need to be able to do repeated bouts of contracting, relaxing, and that will only be possible if we can remove the acetylcholine rapidly from the cleft. And this is the enzyme that allows cells to do that. It's in the extracellular space, but it's linked to the basal membrane using a helper protein, CoQ. And what acetylcholinesterase will do then is it will grab some of the acetylcholine molecules and then break it down into two subunits, choline and acetate. Acetate can diffuse away from the cleft, but the choline, remember I said, was something that wasn't all that commonly used in cells, and so the cell wants to be able to have a way to recycle it. It does that using a choline transporter to reabsorb the choline. This is a symport in which sodium moves down its electrochemical gradient in order to pay to move choline in, and this will allow the cell to rapidly recycle the choline. The choline can then be combined with fresh acetyl coenzyme A to make a new batch of acetylcholine. Well, at the beginning of the video, I said that the neuromuscular junction was a target in many disorders. These can affect the motor neuron, they can affect the glial cells, they can affect the muscle cells. They can be inherited or they can be acquired, and they can range from mild to debilitating to fatal. Let me just give you a few examples. There's a syndrome called Guillain-Barré. This is an autoimmune disease which targets the glial cells that wrap around the motor neurons. And if that myelin sheath is compromised, then these cells become less able to transmit action potentials, and this exhibits itself as muscle weakness. Another autoimmune disease acts on the muscle cells. This is myasthenia gravis. And here the body starts to make antibodies that target the acetylcholine receptor. With fewer acetylcholine receptors available to receive the signal, the patient exhibits muscle weakness. There are still other sets of disorders that affect a number of other players. For example, there's a group of disorders called congenital myasthenic syndrome. So these are inherited conditions. They can affect a number of different players, including cholinacetyltransferase, CAT1, and CoQ. And each one of these, again, would lead to muscle weakness. Finally, there are nerve gases, exogenous agents such as pterin and VX, which can block acetylcholinesterase action, and these can prove fatal. If you continue in medicine, you will undoubtedly see the neuromuscular junction again. So I hope this introduction has been useful.